because we do have a little time. I think we're in good shape. A few questions. We'll do one of those little kind of text it in answer. Let me see if this is still activated for a second. And you can use your phones. You can try to answer these questions. Now, it would probably be a good idea if I was you, looking at the questions I'm writing here, that might be a type of question you would see on an exam. Okay, so don't worry if you don't get it right now, but let's take, whoops, I don't know why it wouldn't just show all the answers at once. Oh, I know what I have to do. Let's see, it always does that. Okay, if I put it in slideshow, there we go. Okay. Whoop. Okay, so I guess, yeah, you guys know how to do it, or scan the thing, or just text to this number, 22333, text my name, 697, and then you can answer this question. Okay. So the last one says microglia, or microglia, I don't know how to, every time I touch it. Let's see, yeah, there we go. I think those are the 5 answers. But one of these is correct. I don't know if there's any more down there. So this type of glial cell myelinates neurons in the central nervous system. Okay, so obviously a couple things there. You need to know glial cells. There's several of them. You need to know what each of them does. Some of them are in the PNS, some are in the CNS. And then you'd have to know what myelination means. We talked about that last time. Okay, this is one of those, you just know it or you don't. You may need to just make a note. Hey, I need to review this. These are so remember our glial cells are our support cells. They're not the neurons, they're the other cells. Myelination, let's just do another aside. Who can remember, what was the purpose of myelinating a neuron? What does that do? It covers the axon of the neuron.
Let's let the electrical impulses jump between. To make it faster?
Faster, yeah. Okay, so we do get that salta, right? The saltatory conduction, which is much faster when a neuron is myelinated. It conducts much faster. It protects it a little bit, insulates it. All right, let's see how we did. So we should see responses here. Hello. Let's see, that didn't work. Okay, good. So most of you chose oligodendrocytes, okay? Which were the cells that myelinate in the peripheral nervous system? The Schwann cells, okay? So if I had written PNS or peripheral nervous system, it's the Schwann cells. Oligodendrocytes, most of you got it here. The microglial cells are immune cells. Those are going to be cells that are just kind of macrophages. They're cleaning up any debris or any foreign microbes or anything like that. So they're kind of, they kind of connect to the immune system. They're going to be those sort of surveillance cells that look around for anything that doesn't belong. Astrocytes, we said, were cells that they help connect neurons to one another and to blood vessels. They surround the blood vessels also and form part of that blood-brain barrier, protecting, not just letting anything flowing in your blood into the brain. You gotta get through these cells first. And epidermal cells we're going to talk more about. We listed them. We're going to talk more about them in the next chapter. They line these fluid-filled chambers deep inside our brain called ventricles. And that's where we make cerebrospinal fluid that we'll get into later. So the answer here was oligodendrocytes. All right, now this one shows the correct answer. Okay. All right, how about this? Another name for an afferent neuron is a blank neuron. Afferent. Let's don't call it out or anything. Let's see if you can get it. We actually use this term even in chapter 1 when we talked about feedback loops. Afferent and efferent. Remember?
Yeah.
Okay, so please, if you have questions, stop me. You want me to repeat anything, let me know. Oh, it does show how many are answered. Okay. All right, everybody take a guess. Anybody having trouble connecting or it's not working to text the answer? Okay. Glad this is kind of working here. still haven't fixed this screen yet, I see. Still very bright. Okay, just give it. Anybody else want to put an answer in? Going once? How did that go down to 14? Does it let you change your answer? I don't know if I said it. You can't change it once you put it in. I should allow you to I mean, if you make a mistake, you should be able to change it. Okay. All right. Let's see how we did. So, afferent means, hey, look at that. Sensory, okay? Sensory. Afferent, these are neurons that are bringing information from your peripheral nervous system, from receptors, into the central nervous system. And then we have efferent. What would be the other word for efferent? Motor. Okay, so afferent, sensory, motor. Good. Okay. Yes. Things were going too well. You know, we gotta just. Oh my goodness. I have no explanation for how that. Hey, okay, whoa. Ah. All right, my little picture here. So this picture, pretty important, okay? We're looking at an action potential, all right, on a graph. Good morning. So at which point in the diagram, wait a minute. Hold that thought. We're gonna go over this. It actually we did in the muscular system, but we're gonna revisit this and talk about some slight differences between the muscular system and the nervous system, but it's the same concept. We talked about that action potential, that the neuron comes down the neuron and it's going to release a neurotransmitter, in this case onto another neuron, not a muscle, and it's going to create an action potential. Remember, action potential was caused by just sodium and potassium and these ions moving across the membrane, where we go from a negative charge to positive and then quickly back to negative. So do you remember, what did we call it? What did I ask here? Oh, okay. Well, what do we call this, like between letters C and E, when we're going up? What do we call that? Good. Depolarization. Okay, and what does that mainly do? Because why does it get more positive in the cell? What kind of ions are moving in? Good, sodium ions. So sodium ions are open here, and then what's going to happen at the top of the peak? The K channel's open, the potassium channel's good. So the answer here, what does it say? At which point, so I would say E would be your best answer. Okay, right at the tip of that peak, our sodium channels are closing and potassium channels are opening. And then we go into, what do we call that coming down? Repolarization. Remind me if I use this term. When we come down, look, it comes down the action potential back down to resting membrane potential, right? At negative 75 millivolts, that would be letter A, which is kind of jumbled there. But look, it goes under. Did we talk about, what is that? We didn't cover that yet. Okay, we're going to do that right now. All right, so let's put that aside for a bit. Maybe we'll come back and do some of those questions in a little bit. I think I have a few with that graph, yeah. Yep. Exactly, that is called hyperpolarization, which I'm going to pull up right here. Let's bring up the slides. And I think we left off right about here. We were talking about how myelinated neurons. I was doing my little Dora the Explorer, right?
Yeah.
Santa, jumping. Okay, so our myelinated fibers are going to conduct larger fibers and myelinated fibers. Those are what determine the speed of an impulse. Myelinated fibers and large fibers conduct much faster than small unmyelinated fibers. All right. So this is kind of what we were just talking about, and we've covered this. This is going to be just like we learned in our negative feedback loops, where we have, eventually this will come up. Let me see, I'm gonna try. Let's see, is this gonna work? Let's give it a shot. Oh, wait, maybe I can put it right in there. Let's see if that works. All right, cool. Wait, worked for a second. There we go. Okay, so sensory, there it is. Afferent, we're bringing signals in. We have our sensory neurons, but most of our neurons are in our central nervous system. They're just packed in tight in our spinal cord, in our brain. We call those interneurons. They are completely contained within the central nervous system. So basically, and we're going to talk more about specific pathways later for different types of information, but you know, you touch something with your finger, let's say we touch something sharp or hot, there's going to be a sensory neuron attached to that little touch or pain receptor. It's going to come all the way in, it's going to enter your spinal cord. When it gets in the spinal cord, it's going to synapse with another neuron, an interneuron. And maybe that neuron travels up your spinal cord, it's actually going to go to a part of your brain called the thalamus, and it's going to synapse with another neuron, another interneuron just in your brain. And then we could have several. We could have a whole pathway of connections from one interneuron to another in our central nervous system. But eventually, well, this is an example of a very quick one. If I touch something really hot, it's actually just going to be a sensory neuron comes right into my spinal cord. It's going to synapse with a small interneuron, which is going to immediately synapse with another motor neuron. And that motor neuron is going to come out now and pull my hand away. Okay, we call that a withdrawal reflex. I'm withdrawing from painful stimuli. That is a very, very quick one, but a lot of times, for example, when you touch something really hot, there is a sensory neuron that comes in, synapses with an interneuron right to a motor neuron out, and your muscle is moving. You're pulling your hand away. Now, we'll synapse with other interneurons that then go up to your brain and maybe to the conscious part of your brain. Okay? But just to protect ourselves, we get that quick reflex loop where I'm just pulling my hand back. But, and I think I've said this before, but you don't realize it happens very quick, but your hand is already moving before your brain even realizes what happened. You don't have any perception of that pain. Your hand is already moving before you even feel the pain. It takes longer for it to get up to your brain and alert that conscious part of your brain that says, hey, what did I just do? You know, I probably should be careful and not touch that again. Okay, so we'll get to reflexes later, but this is basically what it is. Sensory, interneuron, motor neurons. Okay, so touching something, sensory neuron into the central nervous system, interneurons, which can be multiple, and then the motor neuron is the neuron that comes out of the central nervous system to the effector, maybe a muscle to pick something up or withdraw. Okay, couple different examples. Did I put another slide here? No. Okay, so this isn't super important, but we always look at a neuron. You see a picture of a neuron, it's going to look something like this. These are very, very common. We call these multipolar neurons. Okay, so just circle that, multipolar, where it's a cell body. What was the other name for a cell body? Soma. Good, soma. It has multiple dendrites, lots of dendrites coming off of it. And then one axon. Notice it's myelinated with Schwann cells. Okay. And then at the end, we have the terminals where we secrete neurotransmitters. So down here it just shows you receptive. Receptive is blue. Conduction, the action potential travels down the axon. And then the secretory region where we secrete neurotransmitters. So that is a multipolar neuron, very, very common neuron. It's the most abundant in the body, okay? We're gonna skip over to this side. Unipolar. Look at this one. The cell body or the soma is kind of hanging out in the middle here. And we have two processes. One has dendrites, okay? If you want to write, these are going to be dendrites, but they're a little further away. Obviously, they're further away from the body. We call it unipolar because the soma here only has one process coming off of it. As opposed to here, it's got multiple dendrites and an axon, so it's multipolar. Unipolar, just one little thing. And then, at this distal end, we're going to have the secretory region, the terminals. Okay? This is very common in sensory nerves. We're going to see like a sensory receptor, that example I gave before, like at the tip of my finger, I got these little nerve endings here. And then there's a long process, and the cell body is actually all the way up right before your spinal cord. It's far away from the dendrites. But that's what we would call a unipolar neuron. These exist, don't worry about them so much. Notice the cell body, the two processes, 1, 2, so we call it bipolar. These are only found in specialized, like in your eyes and ears, special senses, things like that. But don't worry about that. You're going to be familiar with the multipolar and the unipolar. Those are the ones we're going to talk about mainly. All right, and real briefly, synapses, we're going to spend most of our time because most of the synapses in our body are chemical synapses. Just like we talked about with the muscle, the neuromuscular junction. One neuron releases a neurotransmitter. It basically transfers an action potential from one cell to another. A nerve cell to a muscle cell, a nerve cell to another nerve cell. But some synapses can be direct, where the cells actually connect and they have gap junctions. And those ions can move right from one to another and spread the signal. Okay, so directly connected electrical synapse, the ions, instead of causing a neurotransmitter to be released and then reconstituting the action potential on the other side of the synapse, they move directly from cell to cell. You can even write gap junctions in there. These are going to be more abundant in the embryo. Like a lot of these synapses are sort of automatic. But as we learn and develop, we change these synapses. They're going to change to chemical synapses mainly. Okay? Certain parts of our brain where synapses get really, really reinforced, these stereotype movements, which is like, okay, every time this stimulus happens, we do this. Because we're going to learn that all these chemical synapses, one cell can have synapses from thousands and thousands, tens, hundreds of thousands of other neurons. And some of them are stimulating, some of them are inhibiting, and it's like these cells are making decisions based on all the input. But as to what's going on, what is our response going to be? But some responses are so stereotypical, like we always respond the same way. You know, someone shines a light in your eye, your pupil is going to get smaller. Something like that. Okay, where these synapses are just sort of automatic. Those can be electrical synapses. But by far, the most common types are chemical synapses. I'm going to spend most of our time with those. We're not going to talk about electrical synapses really anymore. Okay. Oops. Wrong computer. All right, so this should look kind of familiar with a chemical synapse from similar to the muscular system. We got our synaptic terminal or synaptic knob. You got your synaptic vesicles with neurotransmitter. And instead of this being the motor end plate of a muscle, it's a postsynaptic neuron. So when we talk about a synapse, we're going to have the presynaptic neuron, which kind of is bringing the action potential to the synapse. And then this would be the postsynaptic neuron. Okay, pre and postsynaptic. You can see the neurotransmitter and the synaptic vesicles. You can see the receptors. That doesn't work. Over there. Okay, so there you have it. Presynaptic, postsynaptic. And depending where the presynaptic neuron releases neurotransmitters onto the postsynaptic, we give them some different names. We can have, let me use the picture, I think it's going to be a little clearer. Get the wrong way, sorry. Okay, so let's look here. Axodendritic, okay? This is kind of like what we've talked about. You're going to have the terminals of this neuron coming from wherever, and it releases neurotransmitter onto a dendrite or dendrites of this postsynaptic neuron. Axon to dendrite, axodendritic, very, very common. Some of them can go directly to the soma, to the body. They bypass the dendrites. We call it axosomatic. Makes sense, tells you right what it is. One axon, the end of an axon, to the soma. Some of them go right, what do we call this little area of the axon? Do you remember? The axon hillock or? Trigger zone. Good, that's an important spot because that's where the action potential will sort of start in the postsynaptic neuron. So some neurons can go right here. Now let me try to, before we get to the actual physiology of the synapse, you may have heard, nerve tissue does not regenerate very well. We know neurons are amitotic, they do not divide. We only have very specific areas of brain where we've found that new neurons can form from stem cells, even as an adult. But generally, we have the neurons we have. But the thing that we can alter from our learning and experience and so on is how these synapses are arranged. And which synapses are sort of, trying to think of the word, accentuated, the ones we use more. But let's just say the signal coming from this neuron is some sort of stimulus that happens. And, maybe at first it's an axodendritic synapse. And as we're going to learn, what happens at the synapses themselves is where we start to go from that resting membrane potential up a little bit, because the neurotransmitter binds, it opens sodium channels, sodium comes in, and we start moving up towards, from -70, let's say, resting membrane potential, more positive. We're depolarizing a little bit. Now, if we get to what's called the threshold voltage, we're going to talk about this specifically in a moment, then this neuron's going to fire an action potential. If right here, in this trigger zone, goes from -70, if it reaches to like -55, don't worry about the numbers this second, we're going to get an action potential. So if the stimulus was here, like picture sodium ions going in right here, they got to sort of diffuse and get there. And if there's enough stimulation, maybe from enough of these dendrites, or maybe there's a bunch of action potentials coming down at once, then we get this to -55, this threshold, we're going to fire an action potential down this neuron. So which one of these synapses is going to be more likely or quicker to get that trigger zone to -50 or -55 to get it to threshold? Is it going to be this one over here, or the one right on the axon? Yeah, it's right there. It's like boom. So it's almost like these synapses can change where they connect. It's like, okay, every time this signal comes down, we always have this response, okay? So we're gonna reinforce it by putting the synapse right here. So it's like, whenever this happens, this is gonna happen. We're gonna have this response. We call this sort of changes in our brain, we're gonna talk about this a little more later, called synaptic plasticity or synaptic pruning. I like the word synaptic pruning because you think of like a bush with all these extra branches and you're clipping them off. When we first, let's say, we're learning to do some motor activity, tying your shoe or playing an instrument, playing a chord on the piano or the guitar or something. Think about that. Anybody here play an instrument or ever try to play an instrument? Right? At first, it's like, okay, I tried guitar. I've tried like 3 times. I don't know. I just think I got to give it up. But, at first, like, okay, a C chord, and you have to like put your one here and then on this string, and you're using so much attention. There's so many synapses happening in your brain right there. You're looking at it. You're thinking about this. So this neuron, the neurons that control the motor are like, all this information coming in. Should I do this? Should I do that? Well, how does this finger feel? How does that one feel? Is it in the right place? But after a while, what happens? All you need to do is see that letter C, let's say, a chord, your fingers just go there. It's like you're not looking, a guitarist isn't looking at their fingers to go, or a piano, or, when you learn to type, right? You don't have to stare at the keyboard anymore, you know where they are.
Would that be considered like muscle memory?
It is. It's very similar to the concept of muscle memory. Whenever you're learning a new activity, whether it's a sport or an instrument, yeah, that muscle memory is this pruning of all these other unnecessary synapses. It's just streamlining this. It's almost like we use this word, a motor program in your brain. It's like initiating a program where once you've done it a few times and you practice and you do it, it's like, okay, C chord. And it just initiates this program and gets rid of, I don't need any visual data coming in after that. I don't need any auditory, I don't need anybody telling me what to do with my hands. Like, it's just, I see the letter C, boom, that synapse came in, I saw letter C, okay, whenever I see that letter C, this muscle and this finger is going to move that way, exactly. And it's streamlined. Synaptic plasticity, that's how our brain changes. We don't grow new cells, but the synapses can alter. Anyway, just know these terms right now, and they're very descriptive terms. So this one we called, I didn't say the word before, axo-axonic. It's a little redundant, but axon to axon. Axon to body, axosomatic, axon to dendrite, axodendritic. Just know what those terms mean. We'll talk a little more about plasticity in a moment.
Question.
Yes?
So the axon to axon, that's the example of plasticity.
That would be, an example is, well, maybe we started with a synapse that was wrong computer. Okay, that was axodendritic. But we've reinforced that so many times, like, okay, every time that happens, we always fire an action potential here. There's like no decision making anymore, because you could have a synapse, we're gonna see this in a minute. I could have a synapse there, and maybe on that dendrite is an inhibitory synapse, telling it not to fire, bringing it further away from threshold, even lower, more negative. And they kind of battle it out. You could have a cell with thousands of synapses. Some are depolarizing, some are hyperpolarizing, and it's like, okay, if there's enough stimulating stimuli, yeah, then we're gonna get an action potential. This is saying like, oh, let's just move from there, because every time that happens, that signal, we always fire an action potential here. So it moves from there to here. So we can make these little wiring changes. Not new cells, just changes in the synapses. Okay, so let's get to the sort of real nuts and bolts here of an action potential, and what's happening at the synapse, and then what happens once we reach this threshold voltage. Okay, so we're going to use the number, although it's not always exactly the same, but most neurons somewhere around negative 70 millivolts. It is more negative inside the cell, like this is the axon, okay, inside than outside. Same as a muscle. Muscle is a little bit more negative, like negative 90, but don't worry about it. So let's talk first about how do we get to this resting membrane potential. Well, we know, first of all, we've talked about this a lot, you learned this in lab, that the concentration of sodium is much higher outside the cell than inside. And the opposite is true for potassium. Okay, much more potassium inside cell than outside. And we have this sodium-potassium pump, which helps us establish that, right? Remember the sodium-potassium pump? It throws out sodiums, 3 sodiums, brings back 2 potassiums. So we can get this gradient here. Now, keep doing that. Okay, the membrane has a lot of what we call potassium leakage channels. They're just, a leakage channel is always open. So if you have a potassium channel open, which way is potassium going to move? It's going to go out. It's going to go down its gradient, natural, just passive diffusion. Okay, it's facilitated, right? But it's still passive. It's just going to leak out. So what's that going to do to the charge inside? Even more negative. Okay, so that if that was the only thing going on, potassium leaks out, it would bring us down to about negative 90, but we do have some sodium leakage channels, a little bit less. Anyway, so some sodium's leaking in down its gradient. That gets us to right about this negative 70, okay? And we use the sodium-potassium pump to make little changes, to adjust it so it gets it right to about negative 70. We need to just redistribute some of these ions. Okay, it maintains the concentration gradient, resulting in the membrane potential, negative 70. Okay, so we're going to look at a chart like this. You're going to get very used to this. Negative 70 is our RMP, resting potential, or RMP if you want to write that in. This is just showing that anything that moves us in a positive direction, we already know that, is called depolarization. So any stimulus that moves us from a negative towards a positive is going to be depolarizing, and anything moves us the other direction below resting membrane potential is hyperpolarizing. We say it's hyperpolarized. It's even more of a difference between inside and outside, not less. Okay?
Say that again. Depolarizing?
Depolarization is where the membrane potential moves less negative, more positive, if you wanted to circle that. So in this direction, we moved up a little bit from negative 70 maybe to negative 65, somewhere there. That's going to be a depolarizing stimulus. We can also, if you want to just write in, this is also called, let me see the next slide, an excitatory stimulus. Excitatory. Why? Because we're going to see if it reaches a certain voltage here, negative 50, negative 55, we're going to get that big spike. We're going to get that action potential. So it's moving us closer to threshold, excitatory. Depolarizing stimuli are excitatory. A stimulus that makes a neuron get more negative is going to be inhibitory. It's going to be, that neuron is going to be less likely to reach threshold and fire an action potential because something made it get even more negative, further away from that threshold voltage. Okay? So just introducing the terms depolarization, hyperpolarization.
What would be like a real example of a
Hyperpolarization? We just call it an inhibitory stimulus, something that's going to make a neuron less likely to fire. We always have situations where we're going to respond to something in a certain way, but there's extenuating circumstances. Oh, maybe I shouldn't do that. Or, I'm trying to think of an example. I want to pick up this $20 bill that I found. Okay, and you reach for it, you do it. Okay, this $20 bill there, you're going to pick it up. But this one's sitting in a bear trap. Okay, you're going to stop yourself for a minute. Your brain's going to wait a minute. I remember seeing on a cartoon that you touch that thing and smack the clothes. So you're inhibiting that response based on past experience. Most of the time this is all autonomic. It could come down to just changing my muscle tone for my posture. Where I'm leaning a little bit this way, so I want to contract some muscles on this side of my body, so I stand up straight. I'm going to inhibit the muscles on this side of my body from contracting, so that I don't fall over. Something like that. Okay, so here we go. Here it is all on one slide. When we have a depolarizing stimulus, we call it an excitatory postsynaptic potential, an EPSP. So let's say we have a synapse between two neurons. This neuron releases a neurotransmitter that causes, let's say, sodium to go in, like acetylcholine that we learned about. That's going to make it less negative, right? Sodium comes in, depolarizes it. That is an EPSP, an excitatory postsynaptic potential. Now we can see this threshold voltage here, and it's about negative 55 in most neurons. So if we can get a stimulus from negative 70 to bring us all the way to threshold, then we're going to fire an action potential on that postsynaptic neuron. Okay? So it's bringing us closer to threshold, depolarizing, excitatory. Put those words together in your head, okay? Depolarizing, excitatory.
Close to the threshold.
Yes, closer to threshold, yes. Now, it's going to take a lot more than just one stimulus. Like, if this was accurate, one little synapse usually would only raise us like half a millivolt or so. So you need several of them stimuli to get us up to that negative 55. We'll see that in just a second. But for now, we're focusing on these terms again: depolarizing, excitatory, and the opposite, there's your hyperpolarizing stimulus. We would call this an IPSP, inhibitory postsynaptic potential. It is hyperpolarizing, below resting membrane potential, further away from threshold. So after this inhibitory stimulus, this neuron's going to be less likely to reach threshold. It's inhibitory, away from threshold, hyperpolarization. I want to just say, so, all right, I'll get to that in a minute. So here's an example. Let's just say here's our presynaptic neuron. It's an axodendritic synapse, okay? So just a regular synapse, and here's our postsynaptic neuron here. So this is an excitatory. It says excitatory, and there it is. And it's not enough on its own to reach threshold. So this is there's all kinds of words for these. It's a subthreshold stimulus. If it was a strong enough stimulus to reach threshold, it would cause an action potential, or we can get several, as you'll see in just a minute. But anything that brings us towards threshold, we say that's an excitatory postsynaptic potential. If nothing else happened, if a signal came, released a little neurotransmitter, it caused some sodium to go in, we spike up a little bit, depolarize, it would just come back down. Sodium-potassium pump would spit out the sodium and bring it down. And it would just have, but maybe another stimulus comes and brings us up again. But watch what happens. If they happen close enough together, see how they're closer together? There's that temporal summation again, tempo. The action potentials come a little bit one after another, a little faster. Now one of these, before it has a chance to come back down, another one comes, and that's enough to reach threshold. Now, usually, again, it wouldn't be just 2, it's usually like 30 or so, right? We gotta go 15 millivolts to get there. If each one of these stimuli only raised us like a half a millivolt, and we gotta go 15, that would be 30. We don't need to know the math there, but it's more than just 2. But the concept here is temporal summation. Think of tempo like fast. One action potential after another. If they're fast enough, they can add up, or summation. They can add up to reach threshold. And look what happens. If we cross this threshold, action potential. This is the action potential above threshold, which we'll review again, even though it's the same as we learned in the muscular system. Okay, so we're just seeing 2 excitatory postsynaptic potentials close together are enough to add up to reach threshold. What about if we have, let's say we have an excitatory synapse here and an excitatory synapse here and a different synapse. They happen simultaneously, but they can also add up because they're both letting sodium ions in. The 2 of them together right here is enough to reach threshold. That's called spatial summation. It's like temporal's time, this is space. Talking about time and space, okay. Okay, so that just means it's another way of adding up this stimulation by simultaneously synapses. So just picture that. We could have hundreds, thousands of synapses happening at the same time. Here's what would happen. What if we had an excitatory synapse, but an inhibitory one? What if they happen, well, here's an inhibitory one by itself. Yeah, we know it hyperpolarizes, brings us away. And it'll come back, it'll find its way back. But then we have at the same exact time an excitatory and inhibitory, they're gonna cancel each other out, right? They'll cancel each other out. And you could have, again, it's hard to sort of fathom when you get to these numbers. It's like talking about stars in the galaxy. But we could have hundreds of thousands of synapses and multiply that by the millions and millions of neurons. You have billions of synapses, trillions of synapses in your brain. I could have a neuron here with, let's just use a random number, 1,000 excitatory synapses and 1,000 inhibitory synapses all happening at once. Nothing would happen. But if they're slightly more excitatory than inhibitory, yeah, then we'll get to threshold, right? If we have more inhibitory than an excitatory, we're never going to reach threshold, which is going to essentially inhibit that signal from moving forward. Yeah.
Does it matter what order they come in?
No. No, they'll cancel out as long as they're within the same amount of time. You know, like these happen simultaneously, they just cancel out. One saying, hey, move up, the other one saying, move down, it's not gonna go anywhere.
The synapse, is it only connects to the dendrites and the cell line?
Yeah, I mean, look, these are showing a very common type, axodendritic. The axon of one goes to the dendrite of this postsynaptic neuron. This one, oh, this one is a somatic, you're right. I didn't even notice that in the picture. This one would be, if you want to label it, this is showing an axosomatic synapse. It's going right to the body. This one's axodendritic. But the concept here is that they are, one's excitatory, one's inhibitory. They cancel each other out. Just multiply that by thousands and thousands, and we could have 1,000 excitatory, but 1,500 inhibitory. More inhibitory, we're never going to get to threshold. Okay, I think I'm going to draw this out a little bit. This is just a picture I found that kind of gives a nice description at the bottom of what's happening at every stage of this process. Let's do this. Good old-fashioned chalk and wax. Okay, let's we have regular white. There we go. Okay, so let's draw a neuron here. We'll do a nice big. I want to leave room for my chart there. Okay, so let's say here's the soma. We'll draw some dendrites. And I'm going to draw it thicker than it normally would be. But a big axon going this way. And I'm going to draw my chart over here so we can kind of keep track of what's going on. Let's say this is our negative 70 millivolts. Let's put millivolts on this side, going up. And this is going to be our RMP, right? Resting membrane potential. Let's make our threshold here. We'll draw this as just a line. We'll say this is negative 55. Okay, this is threshold voltage. And then we'll say, okay, here's 0, and I'm going to go all the way up. We'll say like plus 30 up there. Okay, so I'll just write, inside the cell, it's negative 70. Nothing's happening yet. We're at resting membrane potential. And let's say I'm going to draw another neuron here. We have another neuron here that comes from wherever. And it's going to synapse right there. Maybe we have another one here. Okay, neurotransmitter, right there. And just, you can have lots more of these. I'm just going to draw 2 of them. So what happens is, let's say a signal comes down this neuron, one of these neurons, and it releases a neurotransmitter, I don't know, acetylcholine, and it lets sodium ions in, okay? So what's gonna happen? These sodium ions come in, let's just say a little sodium comes in, it starts traveling this way, it's just diffusing, spreading out, and maybe that will bring us up a little bit. It'll bring us up just a little bit from negative 70. If that was all that happened, just one action potential came down and that was it, nothing else, nothing's gonna happen in the second neuron. It's not enough for it to reach threshold. It's gonna just dissipate, the sodium will spread out, sodium-potassium pump will kick some sodium out, get us back to negative 70 very quickly. But let's say we can do temporal summation. Let's say we send one action potential after another down here, and before this can come down, they start adding up towards threshold, right? So that's temporal summation. Or maybe we have this synapse happening at the same time as this one, and maybe another one over here. A bunch of them together, can also, those can add up, they all happen together. So if enough of these, the sodium ions are flowing in, and those positive charges, I'll just write positives here, they're floating around. And if this is enough to get this trigger zone right here to negative 55, if we reach this point right here, this is what we call the all or none principle. If we reach threshold, we're going to generate an action potential right there in the axon hillock, and it's going to propagate all the way down. You just knocked over the first domino, and it knocks over the next one. It's going to travel all the way down this neuron to the very end. Okay? So once we reach that threshold voltage, we're going to get our spike. Whoa! We're going to get our action potential. I'm going to draw it like that. Okay? So we reach this threshold voltage in the axon hillock right here. We're going to get that spike. We're going to get an action potential right there. So what happens? We have, right in this axon hillock, we have a very high density of voltage-gated sodium channels. Voltage-gated Na channels. There's a huge amount of them there. So, and they're all set to negative 55. If it hits that threshold, they all spring open, and we get a huge influx of sodium. Let me draw it. I've been doing sodium in orange. But, okay, we get a huge influx of sodium right here. Okay, from all directions. Sodium is rushing in there. What does that look like? It looks like this. Okay, so we could say, first of all, we call this depolarization. Going up. We could say, and let's just label. So up here, this is the action potential. These, by the way, these are called excitatory postsynaptic potentials, EPSPs, right? They're excitatory, they're moving towards threshold, they're depolarizing. But we call these, because they're sub-threshold, we call these types of stimuli either EPSPs, or we haven't looked at IPSPs, would go the other way. But we call these graded or local potentials. Local potentials. They're different than action potentials. Local potentials, some could be stronger than others. They bring us up or down a little bit. Whereas an action potential is all or none. If you hit threshold, you're getting an action potential, and it's going to propagate other action potentials of the same strength all the way down the axon. These, some of them could be weaker, some of them could be stronger. They're sub-threshold stimuli, graded or local potentials. If there's enough of them, summation, either at the same time, or multiple, or we have a high frequency, then we reach threshold and we see this. We see the depolarization of the action potential. So right here, if we want to write a little brighter. So right here, let's say, this is where our sodium, I'm just going to write sodium open, but you know what I mean. Sodium channels open right there at that point. That's what causes this mass influx of sodium and this depolarization, where we went from negative inside the cell, past zero, now we're positive inside the cell. As we cross zero and we're up in this region towards the peak of the action potential, you know what happens, right? Sodium, I'm going to write closes, and what happens? Potassium opens. Okay, so right at the top here, sodium channels, their voltage, they have a voltage setting that tells them to open when it gets to negative 55, but to close when it gets positive, like above zero. They start to shut down. We'll watch a little video of this in a minute. Okay, so then with potassium open, we're going to come down this way, which is what? Repolarization. That's the repolarization of the action potential. Woo! Okay, so that way. Now, there's one really important thing that happens here. So let's just see. This action potential happens. Sodium came in, we depolarized. Then, if I can do this, it's like I need to make this into a video somehow. So it went, let's just see what happened. Started at negative 70, it went up to negative 55, and then it went all the way to like positive 30 in there. Now this part of the membrane right here is still a resting membrane potential, right? Nothing happened here yet in the axon. Okay, so this went up, this came back down, but let's just say, okay, it went up to negative 30, and then, I'm just gonna draw it a different color, but let's say, okay, the sodium came in, then the potassium comes out, okay, a large amount of potassium comes out, repolarization, and we go back down. We go back down to negative 70, but notice something very important happens. We don't just stop at negative 70 here, we go below for a little bit. We hyperpolarize, let's say to like negative 90, okay? So it hyperpolarizes, and it's important that it does that, because it's almost like, all right, an action potential just happened here. We gotta repair a little bit. The membrane has to get back to resting membrane potential. It's gonna take a little time, right? This is time on the bottom here, are we done? Going that way. So a little time before it comes back to RNP. Why is that important? So think about what just happened in the trigger zone. We just had an action potential. All this sodium had come in, and it doesn't just sit there. It's going to start spreading around. It's going to, start diffusing, and this part of the membrane, because of all that influx, starts to move up. Negative 7, negative 55. Okay, and it hits, and then we get a spike right there. We get another action potential. Now watch this. Hang on. So this action potential, let's say, just happens. It's happening now. Sodium comes in, right? Sodium, same thing. It diffuses every which way. Now when it goes back here, the area of the membrane that just had an action potential is really negative. It's still hyperpolarized. So which part of the axon, this one's at negative 70, nothing happened here yet. Which direction is more likely to have an action potential, forward or back? This way?
Forward.
This one's closer to threshold.
Yeah.
Look, at negative 70 is closer to threshold than negative 90. It's still hyperpolarized. So this part of the membrane is not going to fire an action potential again. That's what keeps the action potential from going backwards. It only moves forward. Always, we always said action potentials move this direction. That is why. We have this little hyperpolarized period. Another name for that, okay, so let's write hyperpolarization here.
And that's the ISP.
It's not really an IPSP. It is a hyperpolarization, but not due to a stimulus. It's just kind of built into what an action potential is, that it goes a little bit negative afterwards. So that prevents the part of the membrane that just had an action potential from having another one right away, so that it only moves forward in that direction. I'll show you a little video, but we also are going to call this little period I like to think, sometimes they call it, like the membrane's kind of repairing itself. It's getting back to RMP for the net in case another stimulus comes after. But we call this the refractory period. Refractory. Okay? So that little period of hyperpolarization, the membrane's in refractory, it's repairing, it can't be stimulated again. This ensures the unidirectional flow of action potentials downstream, down the axon, not coming backwards. Because the part of the membrane that just had an action potential can't be stimulated again for a little bit of time, and that keeps it moving forward.
So the
Okay,
refractory period prevents the action potential from flowing backwards?
Exactly.
Alright.
That's a simple way of saying it. Yep. Let me, okay, so if you look at this slide, just real quick, and you can see the description here. So here's -70, right? Something during one. A stimulus from a sensory cell or another neuron, whatever, causes it to depolarize towards threshold. So this would be your EPSPs, little local potentials, excitatory, and if there's enough to reach thresholds, if there's spatial or temporal summation, enough stimuli to reach threshold, then we get, if we reach threshold voltage, sodium channels open and the membrane depolarizes completely, followed by repolarization due to sodium channels closing, potassium channels open, but then we get the hyperpolarization as sodium, because the potassium channels, stay open a little bit longer. That's what causes that hyperpolarization. They don't close immediately here. They stay open a little bit longer, so more potassium leaves, we get more negative. And there we go. We call it the refractory period, if you want to circle that. And then the sodium-potassium pump or transporter gets that membrane back to resting membrane potential. But in the meantime, it took a little time so that the signal didn't go backwards. Let's watch a little animation of this. Maybe that'll... Okay, I already muted my computer. What's going on? Let me turn this down. Oh, wait, that's not even volume on here. Let's try this. Okay. Let me just see. Okay, so I'm going to go to the Pearson study area, yep. And again, just like we were doing with muscle, I really highly recommend you, and do the study module, because I built them in there. But if you go to, you can just go to study by chapter, one way to do it, chapter 11. And you can see any of these little A&P flicks or interactive physiology are going to be really good. So let's see, well, I'm just seeing if there's any new IP2s, let's see. Maybe, I might have to go here, hang on. Open another window. Nervous system, where are you here? Okay, resting membrane potential. These are, the IP2s are going to be really good tutorials. Kind of those step-by-steps, asking you questions along the way. But let's just look at one of these first, the resting membrane potential. Hopefully the volume. Okay, now there's no volume. Oh, there.
Contains about 100 billion nerve cells called neurons. A neuron, like every
Can you
other
hear that at
cell,
all?
has positively and negatively charged ions inside and outside.
It's not playing through the speaker.
Further, a resting neuron
Professor?
has a greater negative charge on the
Oh,
inside surface and a greater positive charge on the outside surface. This partitioning of charge creates a voltage difference across the membrane known as the resting membrane potential, which can be measured using a voltmeter. On average, an intracellular electrode records a value of -70 millivolts. The resting membrane potential depends on two factors. First, It depends on the presence of sodium and potassium gradients across the plasma membrane. Specifically, there are more sodium ions outside the neuron than inside, and more potassium ions inside the neuron than outside. Second, the resting membrane potential depends on the differential permeability of the plasma membrane to sodium and potassium
So
ions.
showing some leakage.
Leak channels in the plasma membrane allow
Potassium.
sodium and potassium ions to diffuse or leak down their concentration gradients. The membrane contains many more potassium leak channels than sodium leak channels. Thus, the membrane is much more permeable, or leaky, to potassium ions. As positively charged potassium ions leak out of the neuron, the inside surface of the membrane becomes negatively charged compared to the outside surface. If potassium was the only ion moving, the potential would stabilize at minus 90 millivolts. However, positively charged sodium ions leak into the neuron, which slightly offsets the negative charge and raises the voltmeter reading to minus 70 millivolts.
So we're just explaining the
Sodium-potassium
establishment
bonds
of the resting
actively
membrane
transport
potential.
sodium ions out of the neuron and potassium ions back in, compensating for the sodium and potassium leaks.
Okay, so the sodium-potassium pump just gets us right to that
Thus,
sweet
the
spot.
pumps help to maintain the resting membrane potential.
Okay, we're gonna look at, okay, here we go.
Neurons send signals over long distances by generating and propagating action potentials. Most action potentials originate near the axon hillock of the cell body in the initial segment of the axon. It then travels the entire length of the axon. A closer look reveals that during an action potential, voltage-gated channels open and close, altering the permeability of the plasma membrane to sodium and potassium ions. A threshold stimulus changes the shape of the voltage-gated sodium channels, causing their activation gates to open. This event marks the beginning of phase 1 of the action potential, known as depolarization. As sodium ions diffuse into the axon, the membrane potential becomes less negative. This causes more voltage-gated sodium channels to open, and the membrane
so we're
potential
here.
soars to plus 30 millivolts. At this point, two key events occur. The inactivation gates of voltage-gated sodium channels close.
Don't worry about inactivation gates and things, just know the gates open or close, okay? So if you study this, you know, we know sodium's open, when we get to this peak, sodium closes, potassium open.
And voltage-gated potassium channels open. These two events mark the beginning of phase 2 of the action potential, known as repolarization. As potassium ions diffuse out of the axon, the membrane potential becomes negative again. However, the membrane potential continues in the negative direction, going beyond the resting
Okay, so here we are
of
now.
minus 70 millivolts.
We go below negative
This
70.
marks the beginning of phase 3 of the action potential, known as hyperpolarization. During this phase, voltage-gated potassium channels close and all voltage-gated sodium channels are released from inactivation.
They're still closed.
By the end of this phase, ions move through leak channels only, and the membrane potential
It's gonna
returns
slowly
to
bring
resting
back to
state
resting
of
membrane
minus
potential.
70 millivolts. The neuron is now ready to fire another action potential. Summary. Generation of an action potential. A threshold stimulus.
Okay, so a threshold stimulus, something that gets us from negative 70 to negative 55. So these are like EPSPs happening right now.
opens voltage-gated sodium channels. Sodium ions
Now we're in the action
fuse
potential.
into the axon, depolarizing it to plus 30 millivolts. Voltage-gated sodium channels close, and voltage-gated potassium channels open. Potassium ions diffuse out of the axon, repolarizing it to a negative value. The membrane potential briefly hyperpolarizes.
I mean, I wish they showed this a little bit more going below.
Potassium channels close, and the membrane returns to the resting state of minus 70 millivolts.
Okay, so, you know, take some time to watch these. I think the visualization is helpful. There's one more that just goes into, no, this one. What I was just talking about.
Action potentials propagate
Well, it
in a
only goes
continuous
forward.
fashion in unmyelinated axons. Once an action potential is generated in the initial segment of the axon, it propagates the entire length of the axon.
Okay, one action
Recall
potential after
that a
another.
threshold stimulus causes voltage-gated sodium channels to open. The influx of sodium ions generates an action potential. It also establishes a depolarizing current that flows to the next segment and brings it to threshold. Voltage-gated sodium channels open, regenerating the action potential in this segment of the axon. Current flows from this segment and depolarizes the next segment to threshold, thus regenerating the action potential yet again. In this way, regeneration continues in one direction all the way down to the axon terminals. The basis for unidirectional propagation is revealed when we take a closer look. By the end of the depolarization phase of the action potential, all voltage-gated sodium channels inactivate and voltage-gated potassium channels open. These two events render this segment of the axon temporarily insensitive or refractory
Okay, so
to another
refractory,
depolarizing
we're
stimulus.
and in that negative 90 zone right now.
However, voltage-gated sodium channels in the downstream segment.
So just ahead of the action
are closed
potential,
and
it's still negative
receptive to
70.
Polarizing stimulus. Thus, propagation occurs sequentially down the axon to the axon terminals. In myelinated axons, action potential propagation is a bit different. Here they propagate in a saltatory or leaping fashion. The myelin sheath consists of multiple layers of tightly wrapped glial cell membrane.
Schwann cell.
But this sheath is not a continuous one. Exposed areas of axonal membrane, known as nodes of Ranvier, occur at discrete intervals. Voltage-gated sodium channels are abundant
Same
in the
thing.
nodes, but largely absent between nodes. So, action potentials are regenerated at each node, not in areas covered by the myelin sheath. However, the myelin sheath does provide the insulation necessary for the rapid spread of depolarizing current.
Okay, all you really
And
need
the
to know,
sooner the nodes reach threshold, the faster action.
the myelinated neurons conduct faster, okay? Because it's allowed to jump from node to node. It doesn't have to have sequential action potentials all the way down. Okay, so really highly recommend you do those as well as the interactive physiology. It's gonna really help bring it to life a little bit. All right, so if we go back to these questions, these would be some good examples of a question you might see. So I don't know if will this, I don't know if this will regenerate and you can enter an answer again. Oh, wait a minute, I think I need to, let's put it in slideshow mode. And you should be able to see, there it is. Okay, so if you want to try it, I see 7 people have answered. See if you could scan that, see if you could still answer. There you go, I see more answers, I think, coming in. Went down for a second. Oh, there we go, 8, 9. All right, so this is saying at which point do voltage-gated potassium channels open? Does that code work? Okay, so definitely review this graph, guys, because I can ask a lot of questions about this. All right, anybody else? There's only 10 answers. Everybody take a stab at it. Give another minute. Okay, which point do the voltage-gated potassium channels open?
Let's talk about
Okay, so let's see where we're at. All right, good. So point E, very peak. Said that before. That is where sodium channels are closing. I know it's a little hard to see. That was that's an E. Yeah, not the clearest. I wish I could make it a little bit bigger in there. But yeah, so the top of the peak is where sodium is closing, potassium channels are opening. All right, good. Let's see, what was my next question here? Okay, the points between H and J, what do we call that? There's H, there's J. We could do this one, we just said it. Hyperpolarization, okay, that's going to be hyperpolarization. Also known as refractory period, the refractory period. The membrane is in refraction, it's repairing itself back to RMP so we can then have another action potential. Okay, I'll try to mix in some more test type questions, but we're gonna push forward a little bit here. We got a few more minutes.
Quick question.
Yeah.
What did you say the difference between local potentials and action potentials?
Okay, so local potentials are those excitatory or inhibitory potentials that bring us towards or away from threshold. But an action potential is once you reach threshold, you have an action potential. And just think of it this, one little local potential can happen and then nothing else. It could disappear unless other ones come or they all happen at the same time. But an action potential, once one happens, it's going to trigger another one and another one and another one. It's going to go all the way to the end. All right, let's see what else we got here. Okay, so just some other diagrams. I don't know, whatever works for you. I find the animations, tutorials helpful, but here it is, each step and what's going on at each step. Yep, same thing. I just did another one. It's almost exactly the same.
So when people who have like a lot of salt in their body, like too much
salt, yeah.
salt, like what happens to them?
Okay, I mean, yeah, you can definitely have electrolyte imbalances from salt. You know, when we think about salt, we think more about the water balance. Remember, well, in my lab, I always used to say salt sucks. So like a saltier solution, and not necessarily just salt, I'm just saying a solution that has more electrolytes, more solutes, osmosis, right? It's going to pull water towards it. So that can, like too much salt in your diet, you know, can cause you to accumulate fluid. You know, you're accumulating too much fluid, you're swollen, it can increase your blood pressure because you have too much fluid in the blood, those sorts of things. But electrolyte imbalances, if you have too much or not enough sodium or too much potassium or an imbalance, you can see how that can affect your muscular system. Same thing with calcium. And when you get to cardiac muscle next semester, you're going to see calcium is even more important. Instead of sodium coming in and causing that action potential, it's calcium. And calcium imbalances make your heart start beating really fast, things like that. My daughter called me from college the other day. She's like, you know, my heart's beating really fast and I feel, you know, like I'm going to have a heart attack. I'm like, okay, you're 19 and, you know, I think you're okay. But, you know, I was talking to her, like, did you eat today? No. Like, okay, it's like 2 o'clock. Maybe you should put a little food in your stomach. Yeah, next thing I checked, and I was like, she's out. Okay, So again, just diagrams. This goes through every step by step. If you want to just look at it this way, what's happening at each step of the graph in the top right. So I would say it's pretty important to know. We've only did like 30 slides on this. Okay, same thing. Oh my goodness, beating a dead horse here. Okay, so let's jump to the next little part here. We'll just wrap this up for today. Different synapses can have different effects. We talked about the concept of an excitatory synapse. We've used excitatory, we talked about the neuromuscular junction. Obviously that's an excitatory synapse. But there's going to be lots of inhibitory synapses too. Let's look at a couple of examples. The first one, called an excitatory cholinergic synapse. Cholinergic synapses use acetylcholine. So that is a perfect example, is the neuromuscular junction, okay? All that happens, you already know this, action potential gets there, calcium goes in, causes the release of the neurotransmitter, goes across the synaptic cleft, binds to a receptor, we get some EPSPs, excitatory, reaches threshold, action potential spreads. Okay, that's it, very simple. We've already covered that. This is exactly what happens at the neuromuscular junction, but very, very common neurotransmitter. This is a very common type of synapse. Okay, binding to a receptor directly opens ligand-gated ion channels. We get that depolarization, repolarization. Let's look at another. Let's look at an example of an inhibitory synapse. This one uses a neurotransmitter called GABA. It's a modified amino acid, gamma aminobutyric acid. Don't worry about it. It's just called GABA. Very common neurotransmitter in the brain. Inhibitory neurotransmitter. Why? It goes across the membrane, same thing, binds to the receptors, but doesn't open sodium channels. It opens chlorine channels. Ooh, where am I? Chlorine is negative. Okay, so chlorine, just like sodium, there's more of it outside the cell than in. We open up chlorine channels, chlorine goes in. What does that do to the charge? Does it bring it closer to threshold or further away? Further away, it's negative. So now negative charges are coming in, and that is making it hyperpolarized. This neuron is going to be less likely to reach threshold and fire an action potential. See how that would inhibit it? It's a neurotransmitter that says, hey, move away from threshold, so that even if other stimulatory synapses happen, you're further away from threshold. You're going to be less likely to reach threshold. It's inhibitory. There are some inhibitory neurotransmitters that bind to potassium channels. If we open a potassium channel, positive charges leave, we also become more negative. So some work that way. Anything that makes the inside of the cell more negative is going to be inhibitory, more hyperpolarized. Here's finally an example of another very common type of synapse using a different neurotransmitter, in this case norepinephrine, otherwise known as adrenaline. Okay, so when we get to the sympathetic nervous system, fight or flight, this is the neurotransmitter we use. This one, all I want you to know right now is it works a little bit indirectly. Remember we talked about the second messenger system? Where, let me see, do I have a diagram here? Yeah, so you have this neurotransmitter binds to a receptor. It doesn't immediately open sodium channels, ion channels. It's going to cause the activation of a protein inside the cell called the G protein, which then activates an enzyme, forms a second messenger, in this case called cyclic AMP, and then that can have multiple effects in the cell. We see a lot of hormones work this way. As a matter of fact, norepinephrine is virtually the same as adrenaline. Adrenaline is just if it's in your bloodstream. It's a hormone. But it can open up channels from the inside now. So it's a little bit of a kind of indirect synapse. It doesn't immediately open ion channels, but it creates a second messenger inside the cell. Yeah.
So the GABA synapse brings in instead of sodium or potassium, it brings in chlorine.
Right, it brings in chlorine, but the ultimate effect is that would make it more negative and further away. Yeah. Yep.
And then
Okay.
excitatory, it brings in G protein. So
Well, excitatory, the acetylcholine or cholinergic just opens up sodium channels and that depolarizes, right? This is an example of what we say adrenergic. Think of adrenaline, adrenergic norepinephrine. NE is the neurotransmitter. All it does, it's going to depolarize because it is eventually going to open sodium channels, but it does it in a little bit of an indirect fashion. And many neurotransmitters work this way. They don't immediately open the channels, they cause the creation of an enzyme or a second messenger inside the cell, which then opens channels from the inside. But the ultimate effect is excitatory because sodium comes in, brings us up towards threshold. Okay, I think we're just about out of time. I will pick up and we're almost done with this chapter. I think there's just a couple more slides. But yeah, just like 3 or 4 more slides.